Okay students, today we are going to discuss about continuous sterilization. There is certain drawbacks of batch sterilization which leads to continuous sterilization, because whenever there is any drawback of certain things, we obviously go with another process. So there is major drawback with batch sterilization that is sometimes due to high temperature there is a chance of denaturation of substance which is present in the substrate. So in order to avoid that, batch sterilization is done, and at the same time, continuous sterilization is done. At the same time, whenever the batch sterilization is done, a limited volume of media can be sterilized, but here high quantity or high amount of media can be sterilized and the medium quality can be maintained. But the major drawback with this is sometimes the medium gets liquefied or diluted. Otherwise, this is good because you are saving the power and you are going to sterile a huge quantity. And as I said earlier, the basic difference between sterilization and pasteurization is complete removal of microorganism, whereas pasteurization is only removal of unwanted or pathogenic microorganism. Okay, now we are going to discuss about plate heat exchanger. This is a type of continuous sterilization process. So here in a plate heat exchanger, this is a simple process where how the sterilization is taking place. Just assume that there are 3 towers which is placed for sterilizing for proper mixing. So the best example is like a ballpoint pen. Say here in a plate heat exchanger, just assume the ballpoint. Okay, here inside the ballpoint, the center place, the cold water or hot water is supplied. The surrounding place between this and hot water is supplied, so that the transformation of heat from one place to another may take place, so that in any place where the heat is required, it will be transferred, or in another place when the heat has to be removed, it also can be done. So this is what a best example for any plate heat exchanger. So assume, okay, now in the tower 2, the raw medium is given as an input, so which is passing through the tube this way. Okay, just assume, and it has been passing in the tube and it reaches the third tower. So only in the third tower the steam is given as an input. Okay, so the steam input is given only in the third tower. So here the raw medium is just passed and it has reached. So assume the medium temperature is now 30 degrees centigrade. Once it reaches to the steam, as I said earlier, like plate and chill, this is passing, so the temperature is increased to 70 degree or 80 degree. Now it has passed. So in order to hold for a longer time and to kill the microorganism, the tubes are just curved, so that the liquid will flow slowly along this tube, so that the holding time is increased. As the holding time is increased, the temperature about 70 degrees is maintained for 5 minutes to 10 minutes. Say for this is all for example. So 5 minutes it is maintained at 70 degrees, so that the microbes which is present in the medium or in the substrate gets killed. Okay, so it is all again depends on Arrhenius constant of the microorganism. Okay, then when it reaches the second place, now assume the medium is coming with 70 degree. Okay, this is again an example. Now what you are doing, you are giving an input from 30 degree raw medium. Okay, now this 30 and 40 mixes, so that obviously the temperature of the sterile medium, that is, it is sterilized right now. Once it is passes through the holding section, which means it is sterile. Okay, the microbes get killed. So once this reaches the second column again, the temperature is getting reduced. How means the raw medium you are passing at 30 degree and the sterile medium is now at 70 degree. So that when as in and out layer is passed, the temperature of the sterile medium from 70 as it passes through the column, it reduces to 40 or again 50 degrees centigrade. So just assume 20 degree drop. So the temperature has dropped from 70 to 50. Okay, now just assume, as I said, the input is 30 degree. Okay, as it has been mixed with 70 degree, so once it comes out of the second tower, the temperature from 30, it will be increased to 40 degree. So the input for the third tower is 40. So it is easy to raise the temperature and it is easy to reduce the temperature. Okay, so this is what happens in continuous sterilization process, and once it passes to the first tower, so just you are pouring the cooling water alone. So from 50 degrees, as it circulates inside the tower 1, it reduces to 30 degrees centigrade. So this is the temperature where you can inoculate your microorganism. So it is made ready for the microbial inoculation process. At the same time, why we have been keep on having a plate heat exchanger means in order to save energy. Say once you have been supplying, you need to preheat the medium, and that preheated medium you have to heat it to 70 degrees centigrade. So you need to have an high energy. So once there is a process of heating, no issues, then you need to supply some cold water in order to reduce the temperature. But now this is like 2-in-1 process. You are preheating easily. At the same time, in another tube, the hot sterile medium is coming, so that is get cooled and the raw medium is getting heated up. This happens in each stages, so that you are saving an energy and the liquids and water, everything. So this is what happens in continuous sterilization process.